All right, guys. So I am not here today because my son is sick and I am sick as well. So yesterday we did not finish our organic compounds web concept map, and so I'd like to finish up the polysaccharides area. So if you'll get that paper out, you can follow along. And so polysaccharides are many sugars, and so these are a polymer of monosaccharides, generally glucose. And the first here is going to be called starch, and starch is found in plants, like potatoes or like rice. Those are the things that turn blue when you drop iodine on them, and so starches are found in plants, and that's how plants store their energy. That's how they get long-term energy storage. That's why potatoes are so full of starch, because they're storing lots and lots of energy as these big long chains of glucose. Now, humans have other storage. Animals have other storage uses. They can store their their energy in fats, but sometimes it takes a lot of energy to actually break down and get energy out of fats. We don't break down fats very often, and so if we need fast energy, we use a molecule called glycogen. And so in your muscles, there's a lot of glycogen, and glycogen is another polysaccharide. It's a bunch of glucose molecules, and it's a little bit different structure than starches, but it's found in animals, and it's also used for energy storage. And so glycogen is a little bit less long-term than lipids are, and cellulose and starch are other polysaccharides. Cellulose is the stuff that we burn in wood, and so in plants, cellulose is the fiber and the wood that we actually burn in plants. We don't burn starches in plants, but if you have a wood fire, that is cellulose that's burning, and that's also a stored up energy there. Now what I'd like us to talk about are enzymes, and that's today's lesson. And so we're going to talk about enzymes here. And enzymes are proteins. All right, enzymes are proteins, and their function is to lower the activation energy. It lowers the activation energy of chemical reactions inside the body. And so activation energy means reactions need a certain amount of energy to form, and so they need to increase the energy so that they can release a bunch of energy. And so what an enzyme does is it actually lowers that activation energy. And so when we lower that activation energy, it speeds up the rate of reaction. It speeds up reaction rates. Another way we can talk about enzymes is they are biological catalysts. And biological catalysts are substances that lower reaction rates. And so when we lower the reaction rate of a chemical reaction, it reduces the amount of energy you need for that. We can change the rate of reaction by changing the temperature. If we heat something up, things are going to work a little bit faster. If we cool something down, things might work a little bit slower. We can also destroy an enzyme with temperature. The higher the temperature goes, eventually things stop working. That's why fevers, when they go really high, are so bad. If our temperature goes above about a 105, our body stops working. Our body, especially like 106 or 107, our body shuts down because basically we're cooking all of our enzymes. We're affected by pH. And pH is the acidity, how acid or basic is that environment. We can also change the reaction rate by the enzyme concentration. The more enzymes you have, the faster the reaction is going to happen, because each of those enzymes has an active site that does the reacting. We can also change the reaction rate by the substrate concentration. If there's more substrate, our enzymes don't have to search out the substrate. If it's all right there, the enzymes just get to work. So the more substrate you have, the faster your reaction is going to be. It's going to dwindle a little bit when you have less substrate, because the enzymes have to actively search out those substrate molecules. And so let's go on that. We have enzymes acting on substrates. So those are the things that are being broken down, and the substrates bind to the active site on the enzyme, and that active site is a special site where the substrate fits like a puzzle piece. And so the active site and the substrates bond together to form what we call the enzyme substrate complex. In the enzyme substrate complex, we have the lock and key hypothesis, which basically says that you have a substrate or an enzyme that looks like this, and the substrate is going to look like this, and they fit together perfectly. We also have the induced fit hypothesis. And this one we like this one a little bit better. This is the one that you're going to be tested on. There is another hypothesis, that the induced fit, and basically induced means forced. And so if we have a forced fit, that's basically the enzyme just kind of grabs that substrate and locks the substrate in place. And so we want to focus on this lock and key method, where each enzyme attracts a specific substrate. So you guys should have this filled out, and now at this point your new assignment is to go online to Google Classroom. You should already be there, and you have a reading and some questions to answer in our CK12 Flexbook. Hope you guys have a great weekend, and I hope you don't have too many questions. If you do, we'll take care of it on Monday. Thanks.